Good afternoon and welcome everybody at this academic session in which Ms. Ana Maria Alzate Sànchez will defend her academic thesis in public. Before we start questioning the candidate and giving her the possibility to defend her thesis, she will present to us a short overview of her research and a summary of her most important findings and conclusions. I ask you to start this presentation now and wish you the coming hour much success.
Thank you very much. Dear Prorector, dear members of the Corona family, friends, and colleagues. In the next 15 minutes, I'm going to show you the results of my research. For the start, I'm going to show you about the background. So the main part of my thesis is focused in the basal ganglia. This is a structure that is located under the cortex, and it consists of the striatum, the GP divided into GP externa and interna, the substantia nigra also divided into compacta and pars reticulata, the STN, and the thalamus. The basal ganglia is related to movement, but recent research has also shown that it's related to also cognitive and limbic process. So when we have some fails in the basal ganglia, we have symptoms, movement disorders. One of the most common that is Parkinson. And we also have Tourette, that is one very common in younger patients. These two main groups is going to be the focus of my thesis. So deep brain stimulation is an effective treatment for this movement disorder. It consists of the implantation of electrode which deliver electricity. This and that improving the symptoms of the patients. The good outcome of the DBS depends of the correct position of the electrodes. And for this one, there is different technique. One very used is the microelectrode recording. This technique consists of up to 5 electrodes. They record the activity on the trajectory, and the difference between the activity in the nucleus gave us which is the target. In our case, the STN. So taking this into account, we know the microelectrode recordings are a very useful tool in clinics, but they can be used also to explore different fundamental research. So because we have this research, these recordings in the hospital, we decide to use them to explore different questions in clinical, but also in fundamental research. So first, I'm going to explain the methods that we use. We start from very simple methods and we move until more complex. The first are related to single unit activity. So the first one is firing rate, and this is basically the number of spikes that we have per second. Then we also look to the coefficient of variation. So this what it tells is how much variation there are in the between the spikes. So the higher the variation, the higher the coefficient. And last, we look to the bursting. This is a dynamic state where we have this group of the neuron 5 in groups. So 5 stop, 5 and stop. We then work to a population measurement. The first one is a multi-unit activity. That is basically the total power of the signal. And it combines the single unit spikes, but also the background spike. And the oscillation. the oscillation at this slow rhythmic activity. And as you can see, if we use this envelope of the signal, you can see this mountain-like figure. Each of these is a period. So for measurement of the oscillation, we talk about the number of periods per second, that is a hertz. In our case, we focus in the beta band, which is 15 to 25 hertz. Then moving on to the results, first I'm going to start with the Tourette. So this is an exploratory study. We start with the small group who had only 6 patients, which were all sedated. And what we wanted to look is to see if the anesthesia have any effect. Because of the small group, we just do a very simple analysis, and we found that there was no effect of the anesthesia in the quality of the results. Once we finished this study, we were comfortable with the method that we were using for sorting the spikes. So then we moved to the PD patient, which is a bigger group. In our case, it's 78 groups, patients, which divided into 2 groups. The sedated group, which received anesthesia, in the awake group, they was divided into either the receive anesthesia or the anesthesia was stopped before the recording. So we first looked to the anesthesia. So in the columns, you can see the different agents that they were used. And what we found is that some of them increases the coefficient of variation, so makes it more irregular. And some decreases the multi-unit activity, so the power was lower. We then moved to the patient's variables. You can also see them in the right. So from the old, we found that the disease duration decreases the coefficient of variation, so make it more regular. The hemisphere, so whatever it was right or left hemisphere, it decreases the multi-unit activity. And finally, age also decreases the multi-unit activity. From this study, the fact that the age decreases the multi-unit activity was a really surprising finding, because what is right now in the current research is seems to be the opposite to what we found. So we decided to continue with this. So in the next study, we looked to the effect of age. So as I said before, current research have shown that there is a decrease or a steepening of this 1F slope. The 1F slope, this is a very complex mathematical operation. It basically tells us also the power of the signal. And what the various authors have shown now is that the older patients tend to be stepped, so you can see in gray color, while the black, so the younger patient can see in black, it tends to be more steep. This seems to be different to what we found, and the reason that the authors gave is that there is an increase of noise. However, because most of these studies are done in the cortex with ECOG, there is no way to know if that is correct because they don't have single units. So taking that into account, we use our recordings in the SCN to explore these findings. As we found before, we found this decrease in multi-unit activity. You can see in the older patient there is a decrease in power. As you see, there is less yellow. And we also looked to beta, and we found the same decrease. The next, because of our finding in multi-unit activity seems to be contrary to what we found right now in literature, we wanted to try to explain why this decrease in high frequency power. So with our first idea was there is this increase on noise. And what we did was look to the coefficient of variation and also look to bursting. And we found that there was no difference between the patients. So that seems to be not the answer. Then our second idea was there was a loss of neurons. Because these are doing with microelectrode currents, we didn't have like a direct way to measure the loss of neurons. We used the discovery rate. And what we found is that it was as easy to find single units in the older patient as it was in the younger patient. So this also doesn't seem to be the answer. So finally, we think that there is a decrease in firing rate to explain this decrease in high frequency power. And when we look to the firing rate, we look the older patients have a lower firing rate. So from this hypothesis, this seems to be the one to explain our results. Next, we went to also, because our results were done in the SDN in patients, we looked to a healthy population. And what we found is that in the active, we have a difference between active and rest. So in active patients, there is this flattening of the 1F slope, as it's been shown in the literature. But when we look to the patient in rest, so when they are not doing any activity, we found that is the opposite. So there is this steepening, which is similar to what we found in the STN. And so the answer seems to be there is the difference depends on the mental state of the patient. So in summary, for this study, we found that in the STN, we have a decrease in firing in high frequency, which seems to be explained by a decrease in firing rates. And in the cortex, it depends on the mental state of the patient. So at rest, we have a decrease, while when the patient is active, we found an increase. Finally, in the last chapter, because we have this previous study when we showed that both age and the anesthesia have an effect on the signal, we wanted to look to see if this effect was affecting the clinical. So for this one, we wanted to see if there is an effect of that in the identification of the SDN. So we first start by creating algorithm that will automatically identify the recurrence that in the SDN. We start with a simple algorithm that start with only high frequency, and we have to have a good performance. Based on the current literature, we add beta because they have been shown that seems to increase the performance of the algorithm, especially in the ventral border. So we make a second model with both high frequency and beta, and we found a small increase in the performance. And finally, because we know that there is a difference between the structure that are before of the SDN and after the SDN, we create a model that combines a model algorithm for the entrance and a different for the exit. And this combination is the final model that we have. There is also have a small improvement. So once we have the algorithm ready, we move to look if the anesthesia and the age have any effect. So the first thing that we did was include these two variables into the model, and we found that there was no change in performance. Then we do a cross-training. So we have a testing population and a training population, both either younger or older patients. And when we have a testing population, we found that it doesn't matter if the training population was either the older patient or the younger patient, we have the same outcome. So this seems to indicate that the age and the anesthesia doesn't seem to affect this identification of the SDN. So in conclusion, in the thesis, we show first the sedation does affect the microelectrode recordings. Second, the aging also affects these recordings, mainly by suppressing the activity. But finally, despite this effect of age and anesthesia is not large enough to affect the identification of the SDN. So thank you for your attention, and I give the word back to the rector.
Thank you for this overview, and very well in time. Thank you for that also. Then we can start now with the questioning, the opposition as we call it. And I can mention that actually, apart from Dr. Lowet, who's one of the opponents, all other opponents or were part of the manuscript assessment committee, so I will not mention that each time for each of you. Quite an achievement for the supervisors to have the whole assessment committee here present. That doesn't happen very often. Congratulations with that. And then we start now really this Professor Mess. He's Professor of Clinical Neurophysiology at the Maastricht University Medical Center. He was, oh no, I would not mention all these committees.
Okay,
The floor is yours.
okay, thank you. Well, dear candidate, during the last years, I often saw you walking along the hallway of our department, and you were always obviously absorbed in thought, and that was completely in line with proposition 9 of your thesis, I understand. And of course, I never had any idea that we have a common interest in Japanese writers. Your curiosity has finally led to this thesis, which taught me a lot about MER of the basal ganglia, but maybe that was superfluous because an automatic algorithm will replace all clinical neurophysiologists in the OR, maybe, but maybe that will be discussed on later. So first of all, I would like to congratulate you and the supervisor team with this achievement. Okay, so let's start with the discussion of the meaning of the firing rate and the beta power. Just a simple question, how do age and anesthesia influence these two parameters?
Well,
Very briefly.
Yeah, so for what we have, what we show right now is what they both do is mainly decrease the activity, the multi-unit activity. We know the H does it, and for the anesthesia, we know the dexmedetomidine, that is one that is being used in like more recently, also decreases this multi-unit activity.
Yeah.
For the other, yeah, the effect is not that big. So.
Okay, you just told us so. It's very simple. And I was a little bit confused because in the general discussion, you mentioned that Parkinson's disease is characterized by excessive beta activity in the STN. Shouldn't that mean that age is beneficial for Parkinson's disease? Because with age, you have decreased beta activity. That should be beneficial for Parkinson's disease, right?
Yes, it's possible, but we cannot define Parkinson's disease as just an increase of beta activity, of multi-unit activity. In general, there is other characteristic of the neurophysiological signal that can be in that. I know the recent research, I think for Peter Brown's group, they have showed that is beta in general in power, but also is the burst in beta. The seems also to be related to movement. So we cannot say decreasing beta means automatically Parkinson. There is other things.
Yeah.
So we cannot say that is exactly just because age decrease beta, then it will be.
Okay, this is actually the direction where I want to go to. So my question then indeed would be, do the amount of beta activity and the firing rate actually reflect the disease state of or severity?
Yeah, that's a very good question. I think that is it's a question that I don't think that has been answered yet.
You did a lot of work on this, and you have a paragraph in your discussion where you give the answer to this question. So think about, for example, sex differences, site differences. Is this reflected in the amount of beta activity and firing rate?
For what we have in the moment, no. So when we do the study with the PD patients, we have all these different variables that we look into. We found that none of that affected the multi-unit activity. So there was no difference with sex. Or we also looked to the UPD score, also we didn't find any difference.
Actually, you write that you were surprised, and I was surprised too, of course, when I read it. So would you agree then that the firing rate is not related to the illness severity, but it is a typical pattern of the STN, and it does not reflect proper function? Would you agree to that?
I will say that according to our results, yes, so firing rate doesn't seem to be related to the disease and the disease severity. It might be related to the pattern, but for that we will need more research.
But I think we then have a problem, and you already discussed that problem a little bit in your thesis. And we go back then to the introduction where you have this nice drawing of the box and arrow model. So if it is not the beta activity that is influencing the different parts of this network, could it be that there is something else?
I think it's possible. So we know that the box and arrow is a very interesting, a little bit old model, and it has explained a lot of things, but it's also have been short in explaining other things, which have been explained, for example, with the oscillation model. So yes, it's possible that there is something else, because we also know that there is changes in firing rate when we have like to animal research. We know that once we have the toxin into the monkey, we know that there is a change in firing rate. So we know that that does have an effect on the Parkinson, but might be something else that have to be yet to study.
Yeah, possibly. Okay, thanks a lot for your answers, and I will give back the word to the provider.
Thank you, Professor Mes. Then the position will be continued by Professor Van der Helm. He is Professor of Biomechatronics and Human Machine Control at the University of Technology in Delft, University of Technology. Professor Van der Helm.
Thank you very much, Mr. Pro-Rector. First of all, I would like to compliment you with your thesis. It answers a lot of interesting questions, or raises also a lot of interesting questions. And you do a lot of new measurements, especially the microelectronic measurements are quite unique, I would say. But first of all, I'm looking at the figure 1 in your first chapter, and that is a pleasant figure. If you're from control engineering, then you see a kind of simplified model. But the first question immediately comes into mind: how do you come to this picture? Is this based on electrophysiological measurements, or is this based on neuroanatomical measurements?
Oh, which figure?
Figure 1 in chapter 1, page 11.
Okay. Yeah, so that is based in electrophysiology. That is so it's a simplified version of the box and arrow model. That is the figure that I am.
Because how would the picture look like if it would be based on neuroanatomical information? Because then you obviously has excitatory neurons and inhibitory neurons going from the one structure to the next structure.
Yeah. So it's also a combination. So you have this excitatory, so there's a projection between them, but we also have the change in activity, which is the part of electrophysiology. So
then the picture would become much more complex.
If we add like a lot of details, yes, this is a more simplified version. So it was easy to understand this change, specifically in B, the change there in the different nucleus of the basal ganglia, they had been related to Parkinson.
Because I would think that in your electrophysiological measurements, as you describe in chapter 2 and you measure in chapter 5, then also the spatial resolution of your measurements become important.
I don't understand. Can you repeat the question?
Because you have to, if you measure this if you measure this in microelectrode, your pickup area will be very small. And it can be a coincidence with information you pick up. If you're using your microelectrodes, then the pickup area will be much larger. So then you will pick up a more general signal coming out of it. So how could I relate your measurements that you describe and that you do in respect to this picture, to the neuroanatomical information, which is obviously there?
Yes. So what we have in our in other chapters is the electrophysiological, specifically of the SDN. But yeah, we cannot compare that to see the changes in the SDN related to other structures to know if there is a decrease or increase. That is not something that we can do. We just have the recordings of the SDN.
Yes, because if you do the review of the literature in chapter 2, which I appreciated very much because I think it's quite complete, then you're using the stimulus inputs at the local field potential at the SDN. And then there's some decision time, so there's some processing there, and then there's a response. And then it depends, in my opinion, also on the spatial resolution where you measure the input and the output about what you really can decipher out from the measurements that you describe.
Yeah, so I don't know.
If you measure the stimulus just with one electrode at the SDN, then it's kind of coincidence where you measure that stimulus. And the same count for the response. If you're using just one electrode, then it's kind of in the pickup area is a few micrometers, then it's very coincidental where you pick up the signal, in my opinion. So how do you get to the function of a whole system?
Yeah, we just we didn't go to the function as big general. We just focus in the micro part of the recordings that we have, and if that specific have any effect of other variables, but we cannot look. I think that you will need prospective data to be able to look to the functional part of the general system.
Yes, because in the chapter 5, which I also appreciated very much, because you're doing quite good measurements there, then you're doing, but you describe in chapter 2 is that the input-output relation is depending on the task of the subjects.
In the MEG.
And what were the tasks that you asked for the in the chapter 5 experiment?
Yeah, so for the MEG study, yeah, I'm not really sure. So this was a combined work with the psychology and neuroscience faculty. So the part of the MEG, I'm sorry, I'm not really familiarized because that is not part of my analysis. We know there is a mental task, like a simple cognitive task that the patients did. Well, our SDN patients were just rest, so that they weren't doing anything.
Okay. And then you do the MER processing, and I wondered what you exactly did there. From a signal processing point of view, I had some questions. You first have a high-pass filter of 300 Hertz, and then you're selecting the single spikes. Then I would think then you have the spike rate, which is the information processing. Then you start to do more principal component analysis and another selection mechanism. Why didn't you use the spike rate there as the outcome measure? Because the information is carried on in the spike rate, I would guess.
Yeah, so for the spike sorting, is that the question?
You did the spike sorting, which
yes.
You did personally, so that's kind of difficult. We have to trust you that you did it correctly. But then you selected the individual spikes.
Yes.
And then after you selected the individuals, then I think that you have the individual spikes, then you have the spike rate available. So why did you then do the principal component analysis afterwards?
Because what we needed is just sorting the spikes to the point where we can say a single unit or not. Yeah, and we just focus on more simple analysis for now. So that's why we just get to the point of the sorting, and then we carry some ANOVAs and some statistical analysis.
Okay, because in the end of the chapter you remain to the frequency content of the MER signal. And why did you use that as the outcome measure? And I would, if you have some input measure, you would maybe try to get a transfer function out of it. So how come the frequencies to existence in your signal?
Because there are different, so we sort in, find the spikes, but the frequency, the beta and other analysis was also done in the same, but no sorted. So just after pre-processing, we just calculate the beta without doing the sorting of the spikes.
Okay, good. Thanks very much for your answers.
Okay.
Thank you, Professor van der Helm. Then the opposition will be continued by Dr. Heide. She is working at the Biomedical Signals and Systems Group at the University of Twente. Dr. Heide.
Thank you, Mr. Prorector. Dear candidate, congratulations on completing your thesis, and I would also like to congratulate your supervisors in this achievement. You've done interesting work on the analysis of the microelectrode recordings that were recorded during brain surgery, and yeah, with different goals for each of the studies in your chapters. I would like to discuss a number of steps in your analysis, your signal analysis, a bit further. And starting with chapter 3 and 4, I think most of the analysis was quite similar in these two chapters. First of all, the single unit selection. So you used different radars to select even threshold levels for the spike detection. But there are already quite a number of quantitative measurements or methods described in literature. So why did you use radars to set this threshold level? And also for the clustering, yeah, you already mentioned different criteria might be used by the different radars. So do you know what criteria were used and what should be the optimal criteria for this spike detection and selection?
Okay. So thank you very much for the question. For the first one, yes, so we know that there is some different spike sorting software that is right now. But yeah, with that, we also always sometimes get like really good spikes. Sometimes you don't. So when we start with the Tourette, it was like the first time that we start with this kind of analysis. So that's why we start with the small patients group. And what we did was just kind of trial and error to see how was the best way to sort the spikes. We wanted to do it manually because wanted to make sure that the spikes was done like the best way possible. And yeah, and that's why we start different. First with threshold, we did start first with the automatic threshold. So the code initially will get the threshold, and they will give you the option to pick the single unit that you think are the best. But what we have with that one, it was not always the best. So we have some difficulty to get like really nice spikes with a nice figure and with the autocorrelation, so we can make sure that it was like really just a single unit. So that's why we decided to move to manually. And what we did with that is we did it with different persons. So we were 4 people doing the manual sort of the PD patients. And when we do the comparison, we saw that there was a similar rate in the discovery. So there was a better person doing the sorting than the other one. And even when we look to the spikes, they also look some similar between the different sorters. So that's what we took with these methods. For us, I mean, right now we have this amount of sort spikes, and they have been done by humans, and we have like we know that they are single units. We know they have like good waveform, and they are a good autocorrelation. So we know they are completely single units. And that is something that can be used also to create, a student can use that to create an algorithm to detect bias in this human.
Yeah, you mentioned the autocorrelation function. You use it to confirm the different clustering.
Yes, so we use it to make sure that there was single units. So
yeah, but I would expect, especially if you look at figure 2 or 1, it is in chapter 4. It's easier maybe to select the clusters based on the principal component analysis than using an autocorrelation function. So what would you expect to see in an autocorrelation function? Or how did you maybe instruct the raters to indeed confirm then that it's a single unit?
Yes, so the thing is, yeah, so with just the component analysis, you can get it fairly easy, but with the STN, the thing is, because it's very, very versed the activity, sometimes it's not that easy to separate, and that is something that even with, when we're doing the sorting, we have sometimes this different, because in this you have a very nice image of the two different cluster of activity that can be separated, but we do have in some recordings where the cluster was not that nice, so it was a little bit more difficult to separate. So for that software might be not easy to separate, but that is something that for us as sorter we were able to do it.
Mm-hmm. Okay, then the next step is to use multi-unit activity. I would think that's then the combination of all the clusters. But what you do is you filter the signal and you take the frequency band lower than 500 Hertz, which is usually considered as the local field potential, which is a more global activity compared to the single units, which can indeed be multiple clusters. And so can you comment maybe on using this low frequency band, which you call actually a high frequency band, but still it's more a local field potential than single unit or multiple units?
Yes, of course. So what we did was we have two different types of measures. So we did the sorting so we can do different measurements for single unit activity. So we can look to the filing rate, but also we can look to the coefficient of variation so we can see a little bit of the pattern of the filing rate. And we wanted to look to the multi-unit activity. So that is the total power of the signal. So that's why we have these two. We wanted to see the effect of both because these are the two main things that you look when you are trying to identify the STN. So for the entrance, you try to look this increase in activity, and that was something that you will get with the multi-unit activity. But to separate it from the substantial diagram in the ventral border, you will use the pattern. So because the level of activity is similar, what you use is to see which one is more regular and more irregular to look. Okay. So that's what we look to also to the pattern of the firing rates too.
Okay. Yep. Thank you for your answers.
Thank you.
Give the word back.
You're satisfied. Thank you. Then we continue. Professor Kotz continues. He's professor of neuropsychology and translational cognitive neurosciences at this university. Today he's acting also as secretary of this committee. Professor Kotz.
Thank you, Mr. Prorector. Dear candidate, first of all, my congratulations to this incredible overview. That goes also to your supervisors. It's for all of us who do this type of work, very important results. I would like to ask you a number of questions related to Chapter 4. In particular, I have a few ease-in questions that are simply of interest for me. And one is, do you suspect that there are any consequences of prolonged medication before DBS in patients? And did the patients that were in your sample actually go through a washout before they started the implantation?
Yes, so we know that one of the problems with a lot of Parkinson patients, and one of the reasons that sometimes they come up all the way to the DBS is because the medication start not working, or they have the side effects becomes like really severe. And so we do know that that is a possibility. About the washout, yes. So I think it's 24 hours before the surgery that they are off all the medication before they can go.
Do you think that's sufficient? Because of course one could speculate that there have been alteration in the system that you go in through the long-term medication beforehand. That could be one of the factors that you're not talking about.
Yes, I don't think it's easy to know, because when we go to the patients, we also have to take into account the health. So we know that they have to withdraw the medication so they can go to the surgery, but we cannot ask them to withdraw too much because they will have the symptoms and the quality of life will be not really that good. So we think that there is a compromise and is a standard procedure for DBS is this withdrawal time.
Okay, thank you. My second question relates to the possibility that there may be very interesting data that comes from areas that you target through the stepwise placement of the electrode. For example, would you expect different kinds of changes due to local anesthesia relative to PSA in the thalamus? Have you thought about that? Has there been any data about that? And how informative would that ultimately be for placement in the STN?
Yes, I think that is something very interesting to be able to look to other structure. We did try, but yes, sometimes it depends a lot of the patient. So you have some patients that have to go under general anesthesia. So for that, it's very difficult to take a look and also to have a control because it's similar to what we have with the Tourette. Because the specific of their disease, of the motor and they are not able to control, they have to be under anesthesia for the surgery. So that, for example, we didn't have a proper control. But I think it will be interesting to see with other movement disorders. Because, for example, with Parkinson, we have mainly STN. If we want to look, for example, in the substantia nigra, that one depends a lot of the patient. So in our patient, we do have some patients that the electrode goes all the way to the substantia nigra. So we have maybe 1 or 2 depths there, but not all the patients go there. Some patients just go from outside of the STN to just, again, white matter. So I think it will be interesting to see that with other patients, but we also have to see what kind of patients and how much you have, because with Parkinson, we also have it's a very common disease, so we also have a lot of patients. With other diseases, not all of them will go to DBS.
One additional question, and that has to do with your conclusions of this chapter. So you summarize that patient-dependent characteristics influenced the multi-electrode recordings, but that these effects are not large enough to be clinically relevant. So the quickest answer would, of course, be the sample size, but why do you think these results could be clinically informative? In what way?
So I think that from the patient-related, I think that the most important is age. And I think it's very important not only because we have this effect that it has in the signal, but also we know that age is related to the outcome of the patient. So the oldest patients tend to have a lower positive outcome compared to younger patients. So we think that that is a very interesting line to continue to see if it might be related to some of these changes.
But issues like lateralization or disease severity are probably something rather important, right? Because I'm thinking from a neuropsychological perspective, because sidedness of symptoms can drive very different cognitive profiles in Parkinson's, for example. So in what way would that play a role here? Because it seems it doesn't really pan out.
Yeah, so one of the things that I think there was a discussion that we had early is the fact that we found the UPDRS score that is supposed to reflect the motor symptoms do not have an effect on multi-unit activity. I think that data already tell us that there is a more complex electrophysiological signal and signature to Parkinson than what is being done. And what we found is this disease duration to have this effect in coefficient of variation. I think that is interesting to continue to look to look maybe if it's the pattern of this firing rate that might be also related to the disease.
Okay.
As it is.
Thank you very much. I give the word back to the prorector.
Thank you, Professor Coates. Then the opposition will be contained by Dr. Cubben, excuse me. He is a neurosurgeon in the Department of Neurosurgery at this university hospital here. Dr. Cubben.
Thank you. Dear candidate, also from my side, sincere compliments for the impressive work you did. I'd like to extend the compliments to the supervising committee, but also to a family member, Valentina, who was responsible for the cover art. And my first question is actually going to be about the cover art. Because for a thesis on microelectrode recordings in DBS, I'm missing the microelectrodes on the cover. So is this an omission, or is it maybe a glimpse in the future of DBS? What's your opinion?
Just an omission. I just like this for this cover, like it was. So yeah, we didn't think much about the microelectrode.
Okay, that's slightly disappointing. I hope to the other answer, but that's fine. And so let's continue because I have a few questions on chapter 6, the algorithm. You gave quite some specifics on how you want to detect the border of the STM based on neurophysiological signals, using even different approaches for the entry side and the exit side. And I was wondering, what is the clinical relevance of putting so much effort in detecting the border? Because my philosophy would be, don't bother about the border. Why do you bother?
Well, if we want to implant the electrode, the first thing that you need to know is you need to know the size, so the longest trajectory, which is normally related to what is going to be the end, the implanted, the final electrode. I think it's important to understand both borders so we can have some figure of how, this is the STN, this is the Larius trajectory, and it's possibly one of the best trajectory to choose, or one of the electrodes to choose where we're going to implant. So I think that that's what is important to get these two borders.
I don't agree. Let me explain you why. The border detection you described is mainly the STN part, which is used in Parkinson's disease. The DBS leads, which we use for Parkinson's disease, are mostly directional leads, which means the middle two segments are the most important ones, and the distance between the center of the segment is only 2 millimeters. So the area which is most relevant, it's a maximum of 4 millimeters, which you want, this is what you want to get right. If the STN trajectory is any longer, it doesn't matter that much, in my opinion, because I would say what you need to know is the part of the STN which is certainly STN. And this gray zone at the edges of your algorithm, both entry and exit, won't differ or won't change the implementation strategy. So again, try to convince me that knowing the borders and knowing the exact length of what could be STN would make a difference in clinical strategy, clinical outcome, clinical implant. I mean, that it would be nice for fundamental neuroscience, I agree. That it is nice to develop an algorithm which can do that, I agree. But where is the added value for DBS? The title of your thesis is Microelectrode Recordings for DBS. Where's the added value for DBS?
Yes, I do think that so the main one is to get the first border. But the thing is, if you don't get the ventral border, you might end that with like a really short trajectory in a really like the back part of the STN. So the idea is because
why is that true? Because you come in, you have some uncertainty both when you enter and when you exit, but there is a middle part in which the signal is quite clear and should be fairly easily to detect if you are in the STN. So knowing that part, which is certainly STN, that's where your most important contacts, the segmented contacts, are going to be. So this gray zone, hey, when you say it's a little uncertain when you enter, to me wouldn't be that important. So why is it important in your opinion?
Yes, I think that the main part is to avoid to get the electrode in like probably like in the ventral part. It's really short because you can enter with the the electrode, and you can enter from the middle part, but also in the back. So if you take with the back part, you might take just I'm in the STM, but you are just 2-3 depths, and that is not enough. What you need to try to get is to the middle part, and for that one you need to know that is the longest part, so you have more depths, which will be also easier after the DVS to choose which is going to be the contact that is going to be stimulated, and that is going to be right for the patient.
Yes, but you can't make the size of the STN any bigger than it actually is when we implant, right?
No, but you can see which is the longest part, so which is the middle part, which would be probably the best portion for implantation.
Talking about how you try to determine the trajectory, I see you try to improve the algorithm by choosing a different approach for when you enter and when you exit. And you do that by saying, I calculate the 50%, so what's the middle? And then from there on, I kind of reverse and use different for entry and exit. But to use this kind of middle approximate, I mean, now it works because you have the data from the expert neurophysiologist. But if you use the algorithm alone, then don't you create an interdependency? Because the algorithm should be able to detect approximately what's the middle to then back calculate which part should have this entry algorithm and which part should have the exit algorithm.
Yeah, so in online, the algorithm don't do that. So we are basing the entrance and the exit, this half bar, with the data we have. But once there is online to use, it doesn't have to calculate this middle because we already have the threshold based on the patient that we have offline. So the algorithm don't have to do this half middle, just going to have a threshold for the entrance and a threshold for the exit. And the reason that we have that is because of the SDN. So we know that the entrance is, you normally will go from white matter to the SDN, so that is easy. But when you go to outside, you sometimes go from the SDN to white matter, sometimes you go to the SDN to substantia nigra. So it's not that homogeneous as the entrance.
I agree. So in all cases, the good news is you would be able to detect the center of the STN quite well. Now, what I was wondering, you said you didn't include the burst activity in the model because that was computationally more complex. You also say you need an expert neurophysiologist to do this. I was kind of missing the reference over there. Unfortunately, that would be good news for my colleague. But if you look at the signal of the single unit activity, you see increased amplitude, you see the typical burst pattern, which you in this case didn't include in the model, but these two parameters are fairly easy to recognize purely visually. So what's then you didn't include it again, but then compared to this visual inspection of the signal, what's the added value of the model in your opinion?
Well, that's what we want to see when it works in the operation room. But the first thing is we want to make it more objective, because right now it depends a lot of the expertise of the neurophysiologist. And so if we look to our point, maybe we are safe, we have an expert in that part. But our idea with this algorithm is to be able to put it online, that some other centers that don't have an expert can use it. So it makes it more objective, but also more simple to use. And also can be used in other parts. And part of that is because part of that reason is why we didn't include any measure related to single units, because for that one you will have to get additional to the calculation from multi-unit and beta, you will have to have all these sorting spikes, which add also more time. And the idea is not to add more time to the surgery.
Okay, thank you. I give the word back to the provider.
Thank you, Dr. Kupper. Then the opposition will be continued by Dr. Lowett. He's a postdoc at Boston University, USA, in something called Systems Neuroscience or Neuroscience Systems. The floor is yours.
Yeah, thank you, Professor. Very nice thesis, and I want to congratulate for the thesis and congratulate promoters and promoter. And I was especially struck by figure 2 in chapter 5, where you do spectral analysis multi-unit activity in STN, and where you see age-related effects between power and age. So the first question is, so the way the analysis is done, it doesn't give insights about how the beta, for example, and the high frequency band correlates. So whether they share similar information or not. So do you have insight whether the amplitude of, for example, beta and high frequency correlates?
No, we haven't done that analysis yet. I think for now we take in the basic part, so we look to the power of this both. We do have some analysis in the coherence between the electrodes, but I think that the idea will be the latest to be able to look to other characteristics, the beta, and from single unit activity also to see if also it's affected by age.
Okay, that would be really cool. And something else which struck me is that so you see also an effect in the frequencies below beta. And there's been like recent studies showing that, for example, delta can be a stronger biomarker, for example, of Parkinson than beta. So what is your view on this kind of lower frequencies as a potential biomarker, and whether it might relate, and also whether it might correlate, interact with beta, for example?
Well, I don't know if a correlation for with Parkinson. We do know from studies in the cortex that alpha and lower frequency have changed because of age. So it's possible that is because of age that you have these changes, and that is more related to age than Parkinson. Here we just focus mainly in beta of the frequency because for Parkinson is the main one. It's the principal research. And as I would say before, the thing with age is that we know the older patients tend to have a worse outcome. So we've stayed into the beta Parkinson realm because I think it will be interesting to be able to look at that later.
In the Tita, okay. You said Tita?
No. So for now, we just focus in beta in that because one of the things is when it comes to the analysis, it took us some trial and error to be able to calculate beta, because normally when you have EEG or ECOG, you already are in the LFP, so it's easier to go to calculate the beta. So the frequency bands. So for us, it takes us some trial and errors to be able to calculate the beta from these microelectrode recordings. But I think it would be interesting in the future to be able to look in to other bands, because we know, for example, that the ventral area of the STN is related to lower frequency and it's related to emotional processing. There is some research that shows. So I think that would be interesting to look maybe in the future.
Okay, thank you.
That leaves us some time for second round, and Dr. Kebbe has volunteered to have some other questions.
Yes, thank you very much. More generic question. I mean, as I think we now can consider you an expert in reading from the basal ganglia, for besides the brain stimulation, which is a sort of brain writing, we're also considering in the future of neurotechnology, brain reading applications, brain-computer interfacing. So based on your recordings, the microelectrode recordings which you did, what kind of paradigms, what kind of tasks would be suited by recordings from the STN for BCI applications?
Yeah, I think that so if we focus in the dorsal part, we have like the part of movement.
But reading.
Yeah, the question has been asked, and then you are allowed to very briefly answer, finalize your answer, but briefly.
Okay. So from the STN, we know that it's related to movement, and specifically to stop movement. So I think it would be interesting to be able to look to to that part of tasks related to movement, and especially for patients be able, for example, hyperactivity of people, there is no problems to stop a movement. It'd be interesting to be able to see you can research different tasks in the STN.
Okay, that brings us to the end of the possibility for you to defend your thesis. The committee will withdraw and will return in this room in a few minutes to present you with the result of its evaluation. The committee has discussed the quality of your thesis and the quality of your defense. And based on its positive verdict on both, and in view of the previous examinations you passed, it has decided to grant you the doctor's title you seek. The Professor Cemal is entitled to endope you on this distinction. The floor is to your promoter.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Ana Maria Alzate Sànchez, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I will present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Dear Dr. Alzate Sànchez, dear Anna, I'm very pleased to give this laudatio, Anna. And also on behalf of Professor Jason Temel and Mark Roberts, I'm really happy to congratulate you by obtaining this PhD degree. And obtaining this degree really has been an incredible job. And then I'm not just referring to the excellent research you have conducted, but also about the time frame in which you have achieved this. While most students face a delay nowadays for the research and finalizing their thesis due to COVID, it took you only 3.5 years to stand here today. And these 3.5 years, they passed actually really fast, really quickly. And when we go back to the beginning of your thesis, it brings me back to 2017. And at that time, you were still a master's student. And at that time, actually, everything came together because we just started a collaborative research project with the Department of Anesthesiology. And you as a master's student convinced us to hire you and to help us with this project. And then we actually faced a problem. You were good, we wanted to hire you, but hiring you was a challenge because we didn't have the funds to support you. You, however, you really showed to us that you were a hard worker and dedicated to do the job. And from Mark Roberts, you swiftly learned how to analyze and process these electrophysiological data. And thereby, you convinced all of us to provide you with a contract, initially as a research assistant and later on as a PhD student. And then you really officially started in April 2018 with this project. And you really nicely work together with your paranymph, Michiel Bos, on this translational research project, bridging both clinics and fundamental neuroscience. And yeah, so about Anna, so we also just discussed this a bit. And so who's Anna? And we all got to know you as a shy person, quiet person, introvert person. But I think especially today, you also have shown to be really brave. So you conquered your anxieties and you stood here today, give a really nice presentation, and also you answered the questions really excellently. So excellent job, Anna. I'm really proud of you standing here. You're also someone you can really build on and you can trust completely. So all deadlines, all the work, we could blindly trust you on this. Sometimes a bit stubborn, so we also learned to get to know you as a bit of a stubborn person, and you wanted to do the things sometimes the way you wanted it to be done. But in the end, I think this all worked out, and it was really a pleasure to work with you. And I'm also really looking forward, together with our team, to finish the papers, which we still haven't published yet. Today, I also still remember that you came back from Colombia. When you went to Colombia, you met your family and friends again back in Colombia. And when you came back, you regained a lot of energy from there. You talked a lot about your family and also how happy you were to see all of them. And it has been a while again since you have seen them. And I know you're looking forward much to go back to Medellin upcoming Saturday. Querida familia de Ana. Ana ha trabajado muy duro los últimos años. Ana ha hecho un excelente trabajo. Puedes estar orgulloso de Ana. Cuida de Ana cuando vuelva a casa. Ana, te deseo lo mejor en Medellín.
Dear Dr. Sanchez, it is also a pleasure for me to congratulate you on behalf of our university. And I hope that your last words have been understood because you did not push that button. So maybe they are not on the stream. Yeah, but it doesn't matter so much. Yeah, it's nice to have somebody who is not fully from this university, but you have ended your master studies here, I think. So you can consider you an alumnus of this university. And then it's pleasant and good to have you also finalizing your academic training today by receiving this doctor's title and connecting your name forever to this university. Thank you for that. I also like to thank our guests from other universities. To start with Professor van der Helm, Dr. Heida, and Dr. Loet, guests from abroad and other universities, for taking part in this ceremony. It's always a pleasure and an honor to have you around. Thank you for being here. Then we are approaching the end of this academic session. Normally we will leave in a solemn procession direction, refectory, but today we will head to the staircase in the hall as a group for making a picture. We have a photographer, yes, and you may follow us in that procession. And then at the bottom of the staircase, there is a short moment to congratulate the young doctor, and then we disperse, and then everybody goes his way, and that is the end of then this part in this aula. And with these words, I close this academic session.